Finally, the important part. In this section, we'll go through an example of actually using flowcytoscript. For the most part, this is going to involve typing information about your experiment and pressing run. To get started, let's create a folder for our analysis. Now, sometimes you can run into issues with the folder names getting too long if you use Dropbox or OneDrive. So the safest option is to run this locally on your computer, for instance, in the Documents folder. So I've created a folder in Documents for this tutorial. It has 3 things in it. The data that I exported from FlowJo, I put these in a folder called Data, a copy of the script, flowcytoscript, and a copy of the source files folder. Both of those are in that zip file that you downloaded from the Listed Labs GitHub. You're going to need all 3 of these things in a folder just like this in order for the script to work automatically. The data that I'll be using for this example are those 50-color mouse data from the lymph node, spleen, and gut, gated on viable CD45 positive cells. To start, make sure that you don't have R or RStudio running. If you do, close them down. Then double-click on this flowcytoscript file to open it in RStudio. You should see an interface like this. Menu across the top. The script runs in this section up here, and then we have this console section down here. You're going to be working primarily in the console in the lower left. The script starts off with some instructions for the user up here at the top. I recommend reading these if you haven't gone through the rest of the tutorial. We're going to skip this because this is the tutorial. We'll scroll down to the section labeled 1, Start Up and Install Any Missing Packages. This is the only section where you might modify anything, and you don't really need to if you don't want to. This part of the script controls how much it talks to you. If you've used this script a lot and you want it to shut up rather than telling you things all the time, you can tell it to stop talking by changing the be chatty from true to false down here in this gray section. The gray boxes are the actual code that tells the computer what to do. If the messages are going too fast for you to read, you can increase this delay time here. I have it set to 0 so that I don't have any delay between the messages, just so we can go through this a bit faster. All right, we're going to run this first section. To do this, we're going to click this green triangle run button over here on line 36 in this gray box. When we do that, we get text appearing in this top and bottom section. To see what it says, we can scroll down and read it all, or we can scroll upwards in this console section or expand this upwards. Okay, this is just telling you what the script is going to do. This section is going to install any code packages that you need to actually run the script, things that you need to do, t-SNE, UMAP, clustering, statistical analysis, graph the results. This should all happen automatically, and you may get some red text telling you it's working on that. In my case, I didn't because it's already done the installations. If you get anything that says error, you may have an issue. If that happens, try to close everything and start again. If it happens again, reach out and we'll see if we can help you. And like I said, if you've run it once before, basically it should run very quickly, and you should just get this message saying, that's all done, let's go on to the analysis. All right. In the second section, we're going to run some checks to make sure everything's okay before we go on to doing your analysis. So again, we're just going to click this green arrow on the second chunk. It's going to do some stuff. The computer is going to talk to you. That's where we get these red messages. This is okay. All it's telling you is some information about what it's doing. It's loading stuff. It's attaching stuff. There's no errors or warnings here. Okay, this is all what it looks like when it's working properly. We're going to go down to section 3. Define the experiment. This is the part where we actually need to start telling the script what to do. Okay, I'm going to click this green triangle on line 66, define experiment, run this chunk. What we get is, again, it's telling us, I need your input. First off, how many groups do you have? Well, in my case, I have 3 groups, spleen, lymph node, and gut. I put in 3, and I'm going to put that in as a number. So the number 3, I'm going to put that in the lower part here. So I put in 3, and I hit enter. It's going to ask me for an identifier for each of these groups. Here we need to answer carefully. So we want to look at our data folder. So we'll go to the folder where we have our data, open that data folder, and look at how are these actually called. We want to match this exactly. Mine are LN, SILPL, and spleen. If I put in lymph, it's not going to recognize LN. If I put in gut, it won't find any files called gut because they're called SILPL. I can put whichever one of these I want to be group number 1. I'm going to start with spleen. So I'm going to put spleen in here. I'm going to put it in with a capital S because that's the way it is in my data file, and we can check that looking here. I'm going to hit enter. For group 2, I'm going to do lymph node, and that's going to be LN in caps. And for group 3, I have SILPL, so that's lowercase s, lowercase I, and then LPL, like that. Enter after each one. If I've made any mistakes, I get the opportunity to correct them at this stage. I don't think I did. It looks right to me, so I'm going to hit 0 to skip this step. Do I want to enter different names? Well, yeah, I'd kind of like that SILPL to be called gut, right? I mean, that's better. So let's say I do want to enter different names. So let's say yes. Enter the label I want for spleen. I'm happy with that one, so I'm just going to type spleen again. What I want for lymph node, I can call that lymph node written out like that. And for SILPL, I'm going to go with gut. So each of these I'm typing it in and then hitting enter. And it gives me a summary of how my groups will be labeled. So the first part across the top is the identifier that's going to use to pick up the files based on their file name. And the second part is how it's going to appear in the output of the analysis with the names that I like because they're more aesthetically pleasing. Next, we're going to tell what kind of files we're working with. If you went through and prepared your own transformed files in FlowJo and exported them as CSV files with that transformation applied, this is where you tell the script that you wanted to do that, and you'd select option 1. If you're working with FCS files like I am, select number 2 and hit enter. Now we have the option to select which of these channels that we want. So I have a lot of channels here. I've had to extend the window upwards. In my case, I'm just going to check basically all of them. Generally, when you're doing analysis, I recommend picking the channels that are relevant for your marker, for your cell type. So if you're working with, say, T cells, you might not pick macrophage markers. And if you've already used the markers for gating to arrive at that cell population, you might also exclude those. In my case, I'm going to exclude the viability marker because I've excluded all the dead cells from the analysis, so it's not really relevant anymore. So I'm going to say I want markers 1 through 48. Not 49, but I also want 50. So I can enter that as follows: 1 to 48, comma, 50, and hit enter. It gives me a list of the channels that I've selected, and I can see that they're in the same order, and viability is not on that list anymore. If I want to change that, I have the opportunity right now. I don't want to. I'm happy with that. Next, I have the option to rename the marker labels. I'm going to skip this step, but if you wanted to, you could do things like renaming F480 or TCR-B to TCR-beta. You want to be careful not to include markers like slashes or backslashes or certain special characters in here because that will potentially mess things up. That's why F480 has an underscore. So I'm just going to say I don't want to do this and say none. So my channels are going to be named exactly the same way as they were originally. Right now, it's going to match my data files. So it's looking in my data folder, finding it samples that match those tags, and we can see that it's come up with 3 files per condition, which is what I have. I have 3 lymph node, 3 SILPL, and 3 spleen samples, so that's good. It tells me how many events I have in each group, how many events I have in each sample. This is a check to make sure that you've actually gotten things the way you wanted to before we proceed with the analysis, which is going to be a little bit time consuming. So if you didn't do this correctly, just close the script and start over. Now we need to tell it about downsampling. Downsampling is reducing the number of events we analyze in order to speed up the analysis and also even out the weighting between samples with different cell counts. So for instance, if I have very different numbers in my lymph node and my gut samples. In FlowCytoscript, we sample events per file. That means that if we enter 2,000 here, we take 2,000 cells from each FCS file in our data folder. For me, that would be 6,000 per group because I have 3 samples per group and 18,000 in total. Unless you have a very fast computer, you probably don't want to run more than about 500,000 total events. So I'm just going to run 2,000 today. That'll do this relatively quickly, and we can get a look at the results. I put in 2,000. Now we have a few options about how the outputs that will be generated. These options are here because we may not want to do particular outputs, and the outputs that it's giving you the choice on are things that can be slower to generate than other things. For today, we're just going to run everything. So I want to say, yes, I do want overlays of all my markers. We need to select our groups. If we're working with more than 2 groups, more than 2 conditions, like I have 3 conditions today, we'll get a prompt like this asking me which 2 we want to compare for our T-Rex analysis. This is a method that plots the areas of high and low representation for each group, showing you at a glance which phenotypes are different. I'm going to pick spleen and lymph node. So I will enter 1, 2 for the spleen and lymph node options here. Hit enter. Do I want to run the cross-entropy testing? This is slow, but I'm going to run it today. We're going to say yes. And then please enter a name for your experiment. So I'm just going to say flow cytoscript tutorial. Enter. Okay, it's doing a few things. It's picking up my data, and now it says it's done. We can move to the next section. So we're going to do that. So we did define experiment. We're going to go down and do clustering. Again, we're going to hit this green triangle to run the section. The first thing we need to decide with clustering is which algorithm we use. Now, FlowCytoscript supports phenograph and FlowSom, because these are 2 of the best clustering methods out there for flow data. FlowSom is probably slightly better, but it probably requires you to know how many clusters you want to look for. Phenograph doesn't, so one thing you might do is run the analysis once with Phenograph and look at the results. You can then use the number of clusters it generates as a guide for repeating the analysis with FlowSom if you want. Today I'm just going to use Phenograph, and we'll see probably that the clustering may not be perfect and why we might go back and use FlowSom. This section will take a bit of time to calculate if you have a lot of cells or if your computer isn't very fast like this laptop. So we're going to hit 1 for Phenograph, and it's going to work for a bit. One of the things that happens in this section is we generate a nearest neighbors network of all the cells. That KNN information is used by many of the algorithms, Phenograph, t-SNE, UMAP, cross-entropy. So we run it once in a very fast way at the beginning. That's done. It only took a minute or so. So now we're going to get to the automated naming of those clusters that it's identified. This is a pretty new feature, so your feedback would be appreciated. We're going to try to match the features of these clusters against the database of cell types. To start with, I need to tell it that I'm working with mouse cells. So I hit 1, enter. I also need to tell it where my cells came from. If I have specific tissue sources where there are unique cell types not found elsewhere, I need to tell that to the script. In my case, I'm just using spleen, lymph node, and gut. Those don't appear on this list. So I just select 1 for immune cells. If I had brain, I'd select 1 and 5 like this. But today, I just have 1. So I just hit enter. I also need to tell it if I pre-gated on my cell types, because that'll help it know that it's working with T cells and subsets of T cells rather than leukocytes and all leukocytes. Today, I do have all leukocytes because I picked viable CD45 positive cells. So I'm going to say all 1 and hit enter. I'm going to take a second to plug in my laptop here. Okay, so it said it was plotting some histograms. And now what it's done is it has generated for us a list of clusters with immunological names. It's asking us if we want to rename any of them. So in order to know if we want to rename any of them, we need to look at the outputs. So the outputs, if we go back to our folder where we put our things, we can see now that there's a lot of new folders that have appeared. These are the outputs of the script. The outputs that we're looking for right now are going to be in the density folder. So we go to figure density, and we can see that there's 5 files in here. We have a plot of the bioexponential transforms that have been applied to each of the markers in our data set. This is from the first file only. We have 2 heat maps that come from the cluster identification algorithm, telling us the score of each of the clusters across the bottom, and you can see there's 20 or so clusters, how well these match based on score against the different cell types that are up here on the left. These are all the cell types that are in the database that it's picked up. We have that in two different ways. And then we have histograms showing us the expression of the markers in our data set. And we can have a look at this quickly. So if we scroll over to the left, what we can see here is this is the clusters. Each of these 20 clusters or so is now plotted for each of the markers in our analysis. So the first one, it says that it is naive CD4 T conventionals. Well, if we go across, we can see that that cell type is low for B220, so it's not a B cell. It's low for CD11B and low for CD11C, so it's not a myeloid cell or a DC. It doesn't express CD19. It does express CD3. It does express CD4, and it expresses CD62L. It also doesn't appear to express FOXP3, although the transform here is not ideal. So am I happy with that being a naive CD4 T cell? Yeah, that sounds pretty good. So what we would want to do is go through each of these and check to see how well it works. For instance, our NK cells, let's have a look at that one, this yellow curve here. They're CD11B positive, and they should be CD19 negative, and they are. They're CD4 negative, yes. And where's our NK 1.1? There's our NK 1.1. So they're NK 1.1 positive. So those look like NK cells. So that's what the script has done for this. It's tried to identify the clusters that have been generated based off of the markers that it expressed. And it's not perfect, particularly when you get to very rare populations at the bottom. When Phenograph does some over-clustering, you'll have noisy clusters, and the identification won't work because basically we're looking at junk. So it will assign a name, and that won't be accurate. For today, we're just going to say that these are good. If we wanted to rename them, we could, and that's where we have this section here. We would pick clusters and say, I would like to rename this follicular B cell subset. It's actually transitional B cell type 3. Or it's mistakenly identifying my macrophages, and that's actually my CDC2s, and my CDC2s are actually eosinophils or something like that. That's where you would need to go through and check this manually. For today, I'm going to say no and skip this section. All right, so that's the clustering done. We have now 20-odd clusters automatically identified with what they are. And the next section in 5, really all we need to do is click this green arrow and go ahead and take a break if it's going to be a lot of cells to analyze. In our case, it's not that many cells, and you can see that it's already done some things. So we can have a look at the results that are being generated here while the rest of the analysis runs. So if we go back to our flow cytoscript tutorial folder, we can have a look at the different folders here. So we can see the outputs. So the heat maps are generated first. So we can have a look at that in the figure heat map folder. We get heat maps, the expression of each of the markers by cluster. This is a little bit harder to interpret, I think, than those histograms, at least for me. But it's the same information. We also have heat map based on condition. So we can see that the lymph node expresses lots of T cell markers. The gut expresses some CD8 TRM markers and some myeloid markers. And the spleen expresses a bunch of B cell markers. That makes sense, right? We can see that split up by individual samples, and we can see that the samples are grouped together. And this is the median expression of each marker in each sample. So we have a nice little dendrogram across the top showing us that the lymph nodes group together and are an outgroup from the gut and the spleen. And this makes sense because spleen and gut both have myeloid cells in them. They have DCs and macrophages, eosinophils, and those aren't really present much at all in lymph nodes. All right, so the other readouts we have a histogram folder. This shows us the frequency of those clusters in each of the conditions with arrow bars. So we can see again that the gut has gamma delta T cells, which we know, and there are naive T cells in the lymph node. Because those clusters are automatically named, we don't have to go fishing through our folder to figure out why is cluster 3 up in the lymph node. Is that actually interesting? We get actually a quick readout of what's going on here. We also have whether those clusters are over or underrepresented in each sample in a separate plot here. Okay, we have PCA plots that are generated. In this PCA plot, we're looking at the cluster frequency as the driver of separation on the PCA. We can see that the samples are clustering together by condition, and we can see in this second plot which clusters, are driving that separation. And we can see that the gut separation is being driven by that gamma delta T cell thing primarily. We also have a PCA plot created based on the marker expression, and again, looking at those markers, which ones are driving. Well, it's GATA3 in the gut, that's probably your ILC2s. We have T cells in the lymph node, and we have B cells in the spleen. Oh. We have a table that are generated. If we open these in Excel, we can have a look at the data that are here, which is the frequency of each cluster, so that we can in each sample. And this is useful if you want to then go and plot this in another program, say Prism, make nice graphs that are harder to do for you in R. You can also run your own statistical analysis on this. And this is, in this case, this one is the proportions or the frequencies. The other one is counts. We have stats that are generated. Again, we have CSV files here. This is for each marker. What is the median expression different between the conditions? And it's a summary. So if we have a look, say, at gamma delta TCR, maybe we will see something. Well, let's look at NK 1.1. We can see there's a significant difference between the spleen and lymph node. Now, this isn't terribly interesting when we're looking at total CD45 positive cells, but if you're looking at, say, exhausted CD8s, where you'd restricted the analysis much more tightly at the beginning, you'd say, okay, I have a significant difference in markers A, B, C, and D, and it just systematically runs through all the markers in your set. Okay, the analysis is done. So the other things that have been done in the meantime are we have t-SNEs. And here we have a plot of how well the t-SNE converged. We have our samples, and these unfortunately show up as black in the default Windows viewer. So let's just drag these into PowerPoint quickly. So let's have a look at that B220 plot. And we can see that we have the name of the group across the top, spleen, lymph node, and gut, and we have heat mapping of the B220 on our t-SNE, right? So this is our B cell cluster down here. If we go down here, we can see that we also have plots of our clusters. Where did they go? Here we go. So we have each of those colored clusters that's been generated here, and we can see that each one is a slightly different color. Because there's so many clusters, the colors are actually starting to get relatively close together, but we can zoom in and have a look at this better. And this is, again, split up by condition, and the clusters are named in the legend on the right-hand side. We also down here have the same information, but on all the events plotted together. So we could have a look at this to see how the t-SNE looks in general. And the t-SNE looks like it's worked pretty well. We have nice, well-shaped groups and that are matching relatively well with the phenograph. We're starting to get some splitting here and there. In the UMAP folder, we have the same type of thing. So the UMAP tends to be much more compressed than a t-SNE. That's sort of the only difference is visually. I mean, there's slight differences in the math that are kind of irrelevant for most of us. So if you like the blobs pushed together more, there's your UMAP. What we also have is this changed region plot that I talked about. Here we have statistical analysis between those two groups. So we can look at them for the clusters. So we can say, are the naive T cells distinctly different between the spleen and the lymph node? Because those are the two groups I picked. And yes, they are. There's a difference in frequency between those two that is significant. We have two plots in here. In this case, what we're looking at is the t-SNE data, and we're looking for regions of over or under representation of the two conditions. The spleen is plotted in blue, so where there's blue, it's mostly spleen. Where it's red, it's mostly lymph node. And then we have the same thing for UMAP. So again, blue, mostly spleen, red, mostly lymph node. This at a glance tells you, okay, this region might be interesting for lymph nodes. Oh, this is almost entirely spleen. I wonder what that is. It's probably eosinophils. And then finally, we have the cross-entropy analysis. So that's in these figure t-SNE CE diff and figure UMAP CE diff. We get the same type of readouts in both of them. So we have these cumulative density function plots showing you the distribution of probability of the clusters in this case as on the t-SNE plot. We have the same thing for the conditions and for the samples. And we have a dendrogram showing the relationships between those clusters and between the conditions. So again, we're getting that same sort of relationship we saw with the heat maps of the markers. And we're getting more or less the same thing here where we have the spleen and the gut. They're a little more mixed together. This dendrogram is based off of the cross-entropy on the t-SNE itself. And the other important thing that we get from this is statistical testing. So we can have a look at the statistical testing results for the conditions. So we can say that spleen versus lymph node have a substantially and significantly different representation on the t-SNE plot. The spleen versus the gut does not have a statistically significant difference. The lymph node versus the gut does. Now, these calculations are pretty sensitive to the number of cells that you run. Ideally, you want to be running at least 20,000 cells per condition, and we're only doing about 6,000. So we're not at the point where this is absolutely not significant. We would need to run higher numbers, which would take a bit longer. Okay. So when the script's done, we get this analysis complete message at the bottom. We want to hit save in the upper right here. Upper left. This is going to save an HTML document summarizing our analysis. So if we go to our analysis folder and scroll down to the bottom, we see now that there's this HTML document. It's called flowcytoscript.nb. If we open that up in our web browser, we get this, and it contains a record of everything that we entered. Okay. So all of the outputs that came while we were doing the script are here in a format that's probably a little easier to read. Everything that we entered is here. So if we want to repeat the analysis, all of the information is here and we can repeat it exactly. If we scroll down to the end, we have a results section. And here it gives us a little written summary of what we did, how we did this. So we did it with cluster phenograph. We did 18,000 cells. We found 21 clusters. 15 of those are significantly different between the spleen and the lymph node. And you can go to the results to find out which ones and which channels we used, how long it took. In this case, it took 4.5 minutes. It took us longer to set it up than it did to run the analysis. Here's some of the graphs that we get, and this is just a nice visual summary of the results, so we can have a quick overview of what happened. All right, that's everything. I hope that was clear, and I hope that you enjoy using flowcytoscript. If you have problems or you have suggestions for features that you'd like to see included, get in touch and we'll see what we can do. Thanks.